We always hear about that exciting moment when, a new drug is discovered. But what about all the work that goes on behind the scenes to make sure that same medication is safe, effective, and exactly the same every time you pick it up from the pharmacy?
Yeah, that's a great point. We rarely think about all that goes into ensuring that consistency.
Exactly. And today we are going to do a deep dive into the world of analytical methods in process development.
Ooh, that's a good one.
It's the scientific toolkit that, allows us to see and measure and control all those complex chemical reactions that are actually creating the pharmaceuticals.
Yeah, it's really fascinating. And you, our listener, you sent in some great material that touches on so many different aspects of how drugs are made.
Yeah.
And today we're zeroing in on the specific techniques that scientists use to really keep an eye on and steer those chemical reactions that are really at the heart of making these medicines. So it's not just about following a recipe, right? It's about understanding on a molecular level what is going on.
Yeah, it's about really understanding the process, not just going from point A to point B.
Exactly. And these analytical tools, they're so much more than just quality control at the end of the line.
Right.
They're really crucial guides during the entire development process,
Yeah.
helping scientists understand why might a reaction not be working, how can we tweak it, how can we fine-tune it.
They're kind of like, the GPS
Yeah,
for the whole process.
exactly.
So let's unpack why is it so important to have that intense level of monitoring, on these chemical reactions when we're developing new pharmaceuticals.
Well, it really all comes down to consistency and to control.
Right.
When a pharmaceutical company is manufacturing a drug,
Yeah.
regulatory bodies and patients, you and me,
Right.
we expect every single batch of that medication
Yeah.
to have the exact same composition,
Right.
the exact same potency, and the exact same purity.
Right.
So real-time monitoring using these analytical methods gives us the insight that we need to actually achieve that level of consistency and uniformity.
It's about getting the right amount of product without any unwanted substances kind of sneaking in there.
Exactly. It's about yield, how much do we make, and it's about purity, is it clean?
So it's not just about the destination, it's about that whole journey, making sure that journey happens the exact same way every single time.
Yeah, and making sure we know where we are on that journey at any point in time.
Exactly. Now, we've talked in the past about different reaction conditions like, temperature or pressure. But how do these analytical tools help scientists keep track of all of that, even if our sources don't actually give us like specific details about sensor readings?
So imagine you're baking a cake, right?
Okay.
And the recipe tells you bake it at 350 degrees
Yeah.
for 30 minutes.
Okay.
Now, if your oven temperature is fluctuating wildly,
Right.
that cake might not turn out right.
Right. It might be burnt on one side, uncooked on the other.
Exactly. In a chemical reaction, maintaining precise conditions is even more critical. So while the analytical methods might not directly be measuring the temperature, right? They might not be reading the beige.
Okay.
The results that they give, how much product is formed, are there byproducts present?
Yeah.
Those give scientists a clear indication of whether the reaction conditions, like temperature, were actually properly controlled.
So it's like the cake tasting funny tells you that the oven wasn't right.
Exactly.
I like that analogy.
And if the results are inconsistent, that's a flag.
Okay.
That tells us, hey, there might be an issue with how we're controlling this process.
Now, we've also heard the term kinetics, and that's about how quickly a reaction happens.
Yeah.
So how does the data from all these tools help us understand that we talk about how a drug moves through the body? This is really about the reaction itself. Right?
Exactly. So understanding kinetics in process development is really about figuring out what's the optimal cooking time, right? So you're not undercooking your ingredients or overcooking your ingredients.
I like that, yeah.
So by taking samples of that reaction mixture at different time intervals and analyzing them, scientists can measure how quickly are those starting materials converting to the desired product.
Okay.
And this data helps us to figure out what is that ideal reaction time. Not too short, where you don't get enough product. Not too long, where you might start getting those unwanted byproducts.
So we're essentially creating this, timeline of the molecular transformation.
Precisely. And HPLC, or high-performance liquid chromatography, is a really useful tool for this. I think of it as a molecular stopwatch.
Okay, so HPLC, that seems like a key tool. What does it actually do?
So HPLC is a workhorse in the analytical lab. Think of it as a really, really efficient filtration system, but at the molecular level. So you have a liquid sample from your reaction,
Right.
and you force it through a column packed with these tiny, tiny particles.
Okay.
And these different molecules in the sample, they interact with those particles to different degrees
Okay.
based on their chemical properties. Some stick around a bit longer, some pass through more quickly, and as they exit the column at different times, they're detected and we can quantify them. So this separation lets scientists see what's in their reaction mixture, what are we starting with, what's our product, and anything else that might be in there.
So it's like a racetrack for molecules.
Exactly.
And they all finish at different times depending on, their size and how sticky they are.
Oh, yeah, I like that.
How does that actually help us monitor a reaction, though?
So imagine you're taking samples from your reaction, let's say every hour.
Okay.
By running each of those samples through the HPLC, you can actually watch as the starting material peaks get smaller over time
Right.
as they're being consumed,
Okay.
and then the product peak gets larger.
Right.
You can also identify and measure any impurity peaks that might pop up, so this really gives you a quantitative picture of how that reaction is progressing.
Yeah.
And whether you're getting the desired outcome in terms of yield, how much, impurity, is it clean?
Makes sense. So what about spectroscopy? That sounds like kind of a broader category. But what kind of information do those techniques give us?
So spectroscopy, it's a whole range of techniques that look at how molecules interact with electromagnetic radiation.
Okay.
So we're talking things like light, radio waves, X-rays, all that fun stuff.
Okay.
And different molecules, they have unique ways of either absorbing or emitting this radiation based on their structure, how their atoms are arranged.
Got it.
So by analyzing these interactions, we can actually learn about the identity of a molecule, what its structure is, and even how much of it is present.
And how is that structural information useful in process development?
Well, one example is NMR, or nuclear magnetic resonance spectroscopy.
We have a source dedicated to NMR.
We do, and it's a great source.
Yeah.
NMR can give us incredibly detailed information about the connectivity of the atoms within a molecule.
Okay.
So essentially, it's like a fingerprint for that molecule's structure.
And you can see if you build it correctly.
Exactly, especially if you're synthesizing a complex molecule with lots of, rings and branches and things like that.
Yeah.
You need to be sure that you build it correctly.
Right.
And NMR can provide that confirmation.
It can also help identify unexpected byproducts.
Yeah, even if they're only present in small amounts.
Even if they're tiny.
Exactly, because NMR can reveal structural features that just shouldn't be there if you made the molecule that you intended.
Okay.
And one of our sources mentions how coupling NMR with liquid chromatography, LC-NMR, or even mass spectrometry, LC-MS-NMR,
wow,
that takes this even further, and it allows for really detailed structural analysis of individual components, even within a complex mixture.
So you have a tool that not only tells you what's there, but how it's put together.
Precisely.
That's really cool. What about LCMS? Liquid chromatography, mass spectrometry.
Ah, yes, LCMS.
It sounds like it combines a couple techniques.
It does. It cleverly combines the separation power of HPLC, right, with the identification capabilities of mass spec. So first, HPLC separates all the different components in your reaction mixture, and then each component comes out of the HPLC column and enters the mass spectrometer, which basically measures the mass-to-charge ratio of ions.
Right.
And this is a really precise way to identify each compound.
Because each molecule has a unique mass, right?
Exactly. It's like a molecular fingerprint.
So you're separating everything and then getting a really accurate molecular weight.
Exactly.
For each piece.
And as one of our sources on ADME, or absorption, distribution, metabolism, and excretion technologies, points out, LCMS is incredibly versatile. So while it's really crucial for understanding how drugs are broken down in the body, it's equally important in process development for pinpointing and quantifying all sorts of components, including those impurities that might be forming.
It's like that really sensitive detector for anything you don't want in there.
Yeah, for those unwanted guests at your molecular party.
I saw something that really caught my eye in the material. Continuous flow benchtop NMR. Now that sounds like a game changer.
It is a really exciting development.
What is it?
So traditional batch reactions, they happen in flasks or in large reactors, kind of like a big pot.
Right.
But continuous flow chemistry, that involves running the reactions in a continuous stream through these small channels. And this offers a bunch of advantages.
Like what?
Well, you have much better control over the reaction conditions.
Okay.
You can mix things much more quickly and efficiently.
Right.
And continuous flow benchtop NMR actually integrates NMR spectroscopy directly into these flow chemistry setups.
You can watch as the reaction's happening.
Yeah, you can monitor it in real time as it's flowing through the system.
You can see the molecules transforming.
Exactly.
Okay.
It's really remarkable.
That is really cool. And some of our OPR&D sources highlight that flow chemistry itself
Yeah.
can be really advantageous for certain types of reactions like photochemical syntheses or hydrogenation.
Right, where you might need to precisely control the exposure to light or hydrogen gas.
So you're coupling that with the power of NMR.
Yeah, it's like having a window into the reaction as it's happening.
That's incredible. Now, what happens when things don't go as planned?
Oh.
The reaction stalls, or you see some unexpected products.
That's where these analytical methods really... After you're seeing something form that you didn't anticipate. These tools, these analytical methods, they can give you the clues to actually diagnose what the problem is.
So they're kind of like the detectives.
Yeah, exactly.
So give me a specific example. Let's say we're making molecule Y,
Okay.
but we're getting a low yield,
Not good.
and our analysis shows this mysterious peak that we just weren't expecting.
That's always a head scratcher. So first we need to figure out what is that mystery peak, right?
Right.
So we can use HPLC to separate all the components of that reaction mixture, including that unknown peak. And then we can compare its retention time to known compounds, or use LCMS to figure out its mass.
Right.
And that might help us identify it.
Okay.
So maybe it's an unexpected intermediate that just isn't converting properly.
Right.
Or maybe it's a side product that's eating up all of your starting material.
Right. So it's going down a different path.
Exactly. And then if we use NMR, we can actually get the exact structure of that unknown compound,
Okay.
which could really give us some clues about the chemical pathway that led to its formation. Was a protecting group removed too early? Did a rearrangement occur? You know, all sorts of things can happen.
So it's like a forensic investigation, but at the molecular level.
Yeah, exactly.
And each technique provides a different piece of the puzzle.
Precisely. And once you've identified the culprit, so let's say an impurity is forming because of a specific side reaction.
Okay.
At a certain temperature.
Yeah.
You can then go back and tweak your reaction conditions. So maybe you lower the temperature, maybe you change the order in which you add your reagents.
Right.
Maybe you add an additive that's going to suppress that unwanted pathway.
And then you would use those same analytical methods to check if your changes worked.
Exactly. So has the impurity gone away? Is the yield improving? It's an iterative process of, you know, understanding, adjusting, and verifying.
It's about fine-tuning that entire process.
Exactly. It's like tuning a musical instrument.
You know, our outline actually mentioned some real-world examples from the OPR&D literature.
Yes.
I think it'd be really cool to delve into a few of those
Absolutely.
to see these techniques in action
Let's do it.
and really understand the insights that they provide.
Okay, so one compelling example comes from OPR&D 2013b,
Okay.
which focused on a glycosylation of a complex molecule. And glycosylation reactions, they can often lead to a mixture of different isomers. So these are molecules that have the same chemical formula,
Right.
but their atoms are arranged differently in space.
They're arranged differently. Got it.
Exactly. So in this study, the researchers used HPLC analysis to very precisely quantify the ratios of 4 different isomers
Okay.
under various temperature conditions.
Okay.
And this really detailed analysis allowed them to figure out the optimal temperature
Right.
that would favor the formation of the desired isomer. So it wasn't just that they got more product, they understood exactly how temperature influences that selectivity of the reaction.
So HPLC wasn't just saying you have more.
Right.
It was saying you have more of the right thing.
Precisely.
And it's because of this specific change that you made. That's a really cool insight.
Another great example is from OPR&D 2014a,
Okay.
which describes the isolation of an olefin compound. And while that paper focuses on solvent screening and crystallization for purification,
Right.
you can be absolutely sure that analytical methods, most likely HPLC, played a really important role in determining the purity of that isolated olefin after each step.
It seems like they're being used all along the way,
Yeah, they're essential for every step.
not just at the end.
Exactly. You need to know how well your purification is working.
Right. So you need that confirmation to know that you've separated what you want from everything else.
Exactly. And then in OPR&D 2017C,
Okay.
which details the development of a palladium-catalyzed cyanation reaction.
Okay.
Analytical techniques would have been really critical for this.
How so?
Well, to optimize this reaction, researchers would have needed to assess the yield and the purity of those benzonitrile products that they were making
Right.
under a whole range of different conditions.
Okay.
Different catalysts, different ligands, different temperatures, reaction times.
All those variables.
Exactly. So HPLC and GC, which is gas chromatography,
Yeah.
would likely have been used to actually quantify how much of that desired nitrile was being made,
Right.
and also to detect any palladium impurities that might have been left over.
Because you don't want any of that metal catalyst ending up in your drug.
Right. Right. It's really important to remove those. And the insight gained from this analytical data, it informs the selection of the best reaction conditions
Right.
and the best purification procedures to make sure you get the most product with the least amount of impurities.
That makes sense. What else?
Okay, let's move on to OPR&D 2019B, which describes the synthesis of an imidazole compound.
Okay.
And the authors here, they explicitly state that they used HPLC to determine the purity of their final product.
Okay.
And they achieved an impressive 99.8 area percent purity.
Wow. That's really pure.
It is, and that high level of purity is absolutely critical for a final pharmaceutical ingredient.
Right, because you're putting that into a medicine.
Exactly. They also noted that they saw changes in the color of the solid depending on the isolation techniques they used.
Interesting.
So while that was initially just a visual observation, if they needed to, they could have actually quantified those color differences using spectroscopic methods, which would have given them yet another analytical handle on the quality of their material.
So HPLC gives you that definitive number on the purity.
Exactly, and it meets those regulatory standards.
Right, because everything needs to be documented and proven.
Exactly. And then in OPR&D 2019G, which deals with purification of estrone triflate,
Okay.
they also highlight the use of HPLC to confirm that they achieved high purity 99.1% after a silica gel chromatography step.
So again, it's not just at the end. They're using it all along the way.
Right. It's a continuous feedback loop guided by the analytical data.
And what about OPR&D 2020B?
Ah, yes. That one provides a great example of how analytical methods can be used to troubleshoot specific problems.
Okay.
So during an oxidation step, they found that a diol impurity was forming.
Oh.
And using analytical methods, they could actually quantify the amount of that impurity under different reaction conditions.
So they could see how much was forming depending on what they changed.
Exactly. And specifically, they were looking at changing the quenching agent
Okay.
from sodium bisulfite to sodium sulfite.
Okay.
And by carefully monitoring the levels of both the desired product and that unwanted diol,
Yeah.
they could actually optimize those quenching conditions
Right.
to make sure that the least amount of impurity was being formed.
That's a great illustration of using the data to address a very specific problem in the reaction.
It is. And finally, in OPR&D 2021D,
Yay.
they detail a continuous flow ether synthesis.
Okay.
And in this one, they use GCFID,
Okay.
which stands for gas chromatography with flame ionization detection.
What does that do?
So this technique is used to determine the conversion and yield of the desired product
Okay.
under different flow rates and using an inline separation technique. And this highlights that while we've talked a lot about HPLC and NMR,
Yeah.
other chromatographic techniques like GC, they also play a really important role in process development.
Because there's a whole set of tools,
It is. It's a whole toolbox.
each giving you a different view into that chemical reaction.
Yep.
Now, this all feeds into those crucial aspects of consistency and quality assurance
It absolutely does.
for the final drug product, right?
Yeah, so all that data that's generated by these analytical methods throughout the process development lifecycle, that's really the foundation for ensuring that every batch of drug that's manufactured is consistent.
Right. So you're monitoring and controlling the reaction at every step along the way.
Precisely. And you're building up this really robust understanding of the process.
And then all that data helps inform those quality control tests that are done on the final product.
Right, so that every batch meets the required specifications for identity, purity, potency, all of those critical quality attributes.
And even though our sources don't cite specific regulations,
Yeah.
it's pretty clear that there are rigorous expectations
Oh, yeah.
from regulatory bodies about the quality of pharmaceuticals.
Absolutely. Those expectations are very high, and rightfully so.
Right, because people's lives are at stake.
Exactly. And the analytical methods that we've talked about today, they're essential tools for meeting those expectations.
Right.
So they provide the evidence that the manufacturing process is well controlled
Okay.
and that it consistently produces a drug product of the required quality.
Right.
So it's about ensuring that every pill a patient takes contains the right amount of the active ingredient
Yeah.
and that it's free from harmful contaminants.
So all this analysis, it's not just some academic exercise. It's about, real-world safety and effectiveness.
Exactly. It's about building trust
Yeah.
and making sure that patients are getting consistent, reliable treatment.
This has been a truly eye-opening deep dive into the critical role of analytical methods in pharmaceutical process development.
I agree. It's been fascinating to explore all of this.
From providing real-time insights into these complex reactions
Yeah.
to acting as problem solvers
Right, those molecular detectives.
and ultimately guaranteeing the consistency and the quality of our medicines.
It's amazing how these techniques have revolutionized the way we develop and manufacture drugs.
They really are indispensable. And these tools, they give scientists that deep understanding
Yeah.
of the chemical transformations that are involved in drug manufacturing.
And that leads to more efficient, more reliable, and ultimately safer processes.
It really makes you think, We've uncovered the complexity of developing one single pharmaceutical and ensuring its quality. What does this tell us about the value and the sheer effort behind every single medicine that we rely on?
That's a great question to leave our listeners with.
Yeah, definitely something to ponder the next time you take a pill.
Absolutely. Think about all those unseen hands and all those sophisticated tools that help bring that medicine to you.
And all the science that's happening at the molecular level.
Exactly. It's quite remarkable when you think about it.
Well, on that note, thanks for joining us for this deep dive.
It's been a pleasure.
And we'll catch you next time.
See you then.